Our session is Dr. Sigrid Heuer. She is the science coordinator and program leader for nutrient efficient crops at the Australian Centre for Plant Functional Genomics. Dr. Heuer started her work on Nerica rice with the Africa Rice Centre. Then, as you heard yesterday, did significant work at Erie for a number of years before moving on to Australia. Dr. Heuer.
Okay, thank you very much for the introduction, and thanks, Maurice, for having me. It's my first time in Canada, and I'm enjoying it a lot. Also, the talk so far, very exciting things going on. So hard to beat my predecessor here, but I hope you enjoy also what I have to tell you. I'm going to talk about wheat, which I started working on in Australia, but also about rice.
Stand a little closer to the mic.
Okay, is that better? Yeah. Okay, so this is a rather old slide that I like to show. This was prepared in Erie, and it's showing you the increasing demand and the increase in rice productivity we have to achieve to keep it pace with the increasing demand. And the red bars showing the increase in Asia and the blue bars in Africa. And this is based on data from 2010. So we're preparing for this talk. I thought it's maybe a good idea to update that, and I was quite pleased to see in the FAO database that the global rice production actually is 488 million tons and 494 in 2015. So it's actually exceeding the required increase in productivity, and I like to think that this is probably due to all the efforts going on in rice breeding, so it's actually paying off, and investment in agricultural research is paying off as well. So we need to make sure that this stays this way. So when we look at rice, there are two main production systems, the irrigated, which is generally favorable, and the stress-prone rain-fed environment. And the irrigated system is more productive, so 70% of the rice is produced on about 60% of the area. And this is due because we have constant water control, and with that also some control over wheat. And because it's a safe environment or low-risk environment, farmers also apply fertilizers. In contrast, the rain-fed environment is 40% of the total area in Asia, but only 25% of the production. Obviously, this is due to drought and floods, as I will show you later. But because it's a high-risk environment, also farmers are reluctant to put a lot of inputs, fertilizer or other agrochemicals. It's just because the risk to lose the crop is too high. Also, they don't have the means or the access to fertilizers often. So if you want to make progress in those systems, we really do have to develop stress-tolerant varieties. And at the top of the list is drought stress, but at the same time, because we are in a monsoon region, we're also dealing with submergence stress. Nutrient deficiencies is an issue, but also toxicities, aluminum and iron, which then in the end also causes P fixation and P deficiency, heat, salinity, you name it. So there's a lot of scope to improve productivity in those systems if we have stress tolerance. So the first project I want to talk about is submergence-tolerant rice. Most of you are probably familiar with this project. I don't want to take all the credit, so Dave McKill was the one who started this and also really brought this to the success. And Julia Bailey-Saris in UC Riverside, she did a lot of the science, the molecular and physiological study on this project. So what is the issue when fields get submerged, so completely submerged? The plants try to escape, so they show this leaf elongation, they go to the water surface. And in deep water rice systems, the water stays and the grains can be harvested from boats. But when in submergent-prone environments, then the water at some point recedes, can be several meters of waters for a few weeks, and then the plants lodge, and actually that's the end of it. So what we have identified at the time, or Dave initially, is a major QTL on chromosome 9 that confers tolerance. Oh, sorry, that was too early. And we had the donor variety of this trait is FR13A. It's a landrace, an Aus-type variety, and we sequenced the QTL region, and you see on the top the QTL region in the Tolerant variety, and then in comparison the Nippon Bari reference genome at the bottom. And what we find was we did find 2 ERF transcription factor genes, so ethylene responsive genes, perfect candidate genes, because we know submergence is related to ethylene response. But then when we had a closer look, we found there was a 3rd gene present in only the Tolerant variety, which is absent from Nippon Bari, and that's the sub-1A gene. And this gene, when you have it present, either in transgenics or in this case, introgression lines having the entire QTL, it's actually preventing the escape response. So it's conferring a quiescent status to the plant. The plant is just sitting, waiting until the water recedes. So it's not trying to escape, it's not growing. It retains its carbohydrate reserves for recovery growth after the water recedes. The other plants, as you can see, hugely elongated, and they don't recover. So this is a field demonstration plot in Erie. Those plants had been submerged for 17 days under several meters of water, and then were allowed to recover. And you can see here, if you compare Summer Masori to your right, and you introgress the sub-1 QTL, you can see that the survival rate is much higher. Similar, if you put this QTL into IS64, we have a much higher survival rate. So this is a really beautiful project, and it shows 2 things at this stage. So there's a single gene that can actually have this major effect. Previous to that, submergence tolerance was considered as a very complex trait, so many QTLs involved, and now we know it's just a single gene that can do that. And the other point I want to make here, it's absolutely essential to put those kind of genes or proof of concept into different genetic backgrounds, and we've heard this before, because otherwise there's no adoption, and certainly not in rice, where you have very regional consumer preferences. So this Swana sub-1, Swana is an Indian variety that is very well widely grown, and that was the first sub-1 variety that actually made it into farmers' fields. You can see here, after this field was flooded for several days and repeatedly, there was, in the end, the plants recovered and we had a happy farmer. So this is really beautiful to see this working in the field. Swana sub-1 was tested across many sites, and they had about 1% on average yield advantage over the controls. The third point I want to make with this project is that to really get it out into farmers' fields required a lot of resources, a lot of time, and a lot of money. And Bill Gates at the time came to the rescue, and they pushed Erie to do the seed production. Erie, until then, actually didn't do that because they thought it's in the national systems who should be doing that, and then handed like pre-breeding products or lines to the national system. So that was the first time Erie did this, and Umesh to the variety did a fantastic job mobilizing all the seed producers and get it out there to the farmers. Without that, I think it wouldn't have been the success it is now. The other project I want to talk about is phosphorus deficiency tolerance, and I don't need to tell you that we need to have nitrogen, phosphorus, and partly also potassium applied to retain soil health and to have a high productive system. Again, this project has been started by Matthias Visuva. He's in Japan, and Jirkas was at some point at Erie where we met. Chin is the one who did all the breeding marker development, and then Rico is the one who cloned the gene in the end. So this project started with a screening of a small panel of only like 30 different rice genotypes in a highly P-deficient field in Japan. And Kasalath was identified as a variety that had a much higher P uptake under those conditions compared to, again, Nippon Bari. And then Matthias went ahead to develop a mapping population, so you can see the neosogenic lines C443 in the field. They are productive, they have much higher tilde compared to the Nippon Bari. And there were 2 QTLs identified at the time on chromosome 6, an intermediate effect QTL, and the main effect QTL was on chromosome 12. Again, we went ahead sequencing across the region, and this time Kasalath is at the bottom, the Tolerant one, and Nippon Bari on the top. And you can see again there are huge differences between those 2 genotypes. And the India region is actually 100,000 base pairs which are just missing in the reference genome. And we had a very messy set of 69 pseudogenes or candidate genes, so I spent a lot of night shifts trying to figure out what is actually a gene and what is not. Eventually, we settled on 2 main candidate genes. I'm not going to talk about the dirigent gene because this is actually downstream of the protein kinase. And this protein kinase is the one that we named phosphorus starvation tolerance 1 or PISTL 1. And I just want to mention again that this gene is in the indel, so it's in a unique region specific to the tolerant casellath. When we put it in a constitutive promoter into rice, we screened that in P-deficient soil, and you can see the transgenic plants have a much greater productivity, high yield, and higher tiller number. And if you compare the roots, you can see this enormous root growth of the transgenics compared to the null controls. And because it's a constitutive promoter, we thought we can as well try it in hydroponics. So that's what we did. And even under plus P conditions in hydroponics, this gene is enhancing root growth. So rather than tapping into gene starvation responses, what this gene does is really enhancing nodal root growth or maintaining root growth also under stress. We did a lot of work with markers. We also provided training to national breeding systems to make sure this is getting out there, and the breeding programs can check if they have the gene already or identify suitable donors. And we did screen a set of eerie genotypes. And what we found was, I cannot really point it, but these are the genotypes, and then horizontally, these are the different markers across the region. And the dark color indicates this is the tolerant allele present, and the lighter colors are the intolerant allele. And we found that actually breeders have selected for this QTL already because it's mainly present in the rain-fed rice and in the drought-tolerant genotypes we looked at. It's mainly absent from irrigated rice, I think just because it's not really needed in this type of rice. So at IRI now, PAP1 is being pyramided with other QTLs, and then Matthias at GRCAS, he's still working on this. He worked on this gene in African rice, so we have a different allele in Oryza glaberrima, but it's there as well, so further indicating that this is really an important gene. And field testing of PAP1 breeding lines is still ongoing. Those of you who work on phosphorus may know it's not a trivial thing to really quantify and have statistically significant data to really show the effect. But I think overall this project is, has identified a gene that is really relevant, and the data show breeders already use it actually. The last project and rice that I want to mention is on heat tolerance. This is the variety we use for that is N22. And the graph shows you that the main sensitive stage for heat is during anthesis, so it's affecting pollen development, but also pollen shedding. And I don't want to go into detail, just mention we have worked with Milano University and also with Max Planck to do RNA-seq, and RNA-seq in combination with metabolite studies. And we have now a really nice set of candidate genes that I would really like to see validated, but we don't at the moment have much of a project, and we need to see where this is all going. All those three genes, SUB1A, PISTL1, and also N22, but also genes for salt tolerance, are all coming from one particular group of rice. And this is called the AUS type rice. So this is a SNP marker survey of the different rice genotypes we have. And the AUS type is closely related to indica rice. And the reason why we find all those valuable genes in this group of rice, and I think there are many more really hidden as a real treasure chest, I think, is because this land rices have evolved in problem soil regions. So this map shows the yellow and green dots indicate problem soils. And those varieties are still grown in northern India and in Bangladesh as well. So they're easily crossable. They have a relatively decent agronomic performance. They're actually really good donor materials for those kind of projects. How am I going with my time?
Well, you've got about 10 minutes.
Oh, that's good. Okay. So then can I go back? So just to say, when we have access to 3,000 rice genomes, genome sequences, I think this is a tremendous progress. And then if you can tap into those complex regions and just on the computer pull out the genes that are specific to those kind of regions, that will be just tremendously, so much faster also to get this work done. The problem maybe is that there's a lot of work being done on SNPs. SNP markers rely on genes that are present in all genotypes or most, and probably we're going to miss out on those unique regions a little bit. So that's my concern, but I think this can be fixed. So moving into wheat, this was quite a bit of a shock for me coming from rice, and then realizing that when I started, there was no reference genome for wheat. And look at the size, it's 17,000 megabases compared to 430 in rice. So that's a real shocker. But I think, there's progress there as well, and we have RNA-seq data and survey sequences. So we have resources. We need really good bioinformaticians to help us to pull out genes, but it's moving forward there as well. Wheat in Australia is growing in a stress environment, very similar to what we see in the rice rainfed environment. So the yield is really low, it's 2.5 tons on average compared to 8 tons in Europe. And the main reason for that is because it's water limited. It's always water limited. And in years where we have severe droughts, those are marked with a star, we lose another 50% of this already low yield. But the grain quality is really good, so that kind of compensates it to some extent. Other stresses that are important are heat, same as in rice as well, salt, pH issues, and soil issues, aluminium, but also boron toxicity. And I'm going to talk about some of those. So this is now work mainly being done at the Australian Centre for Plant Functional Genomics. Peter Lang, which was the CEO of this institute for many years, and there's been a lot of work being done on identifying drought-tolerant QTLs. And the variety, there's mainly, or the genotype main used for that is RSC875. This is the one in the middle. It's a really highly drought-tolerant genotype, and a lot of QTL have been identified. And what I'm showing you in the graphic here is summarizing a huge amount of work where all those QTLs have been tested in different environments. And as I said earlier, it's the absolute prerequisite for breeding companies to be interested is that we can show them there's an effect in different environments. And the left side, so QTL 1 to 5, you can see it consistently shows a positive effect, whereas the other QTLs are kind of variable in different years and different sites. And part of the reason for that is you have to be very specific, and certainly with drought, there's no one drought. So you need to know what kind of, what the problem is. And in Australia, surprisingly, when I came, I learned that for drought tolerance, you need shallow roots. And I thought, no, this is really wrong. But it's actually, if you are in a shallow, sandy soil, and you have in-season rainfall, you have to capture all the water before it is, washed away. In other areas, same in Australia or in the tropics, maybe you have deep clay soil, so that's when you need the deeper roots to access remaining soil moisture. Maybe then you don't have rainfall during the season. So to make a progress in drought, number one thing, you need to know what is it you actually need. The center has also traditionally worked in GM drought genes. This is mainly done in collaboration with Pioneer DuPont. And we are still, this is still work in progress. I don't want to talk about this too much, but we are field testing those. Again, this is, we know this, what is hugely expensive and difficult to do. We recently got a significant grant from the Australian Research Council to continue working on this QTL, amongst other things. And we partner now with 3 Australian breeding companies, AGT, Longreach, and Intergrain, and hoping they help us to validate this material further. We also develop genetic populations to work with them. Nitrogen is part of it as well, but I'm just not going to talk about all this. If you want to learn more about this, I'm happy to talk about it. Now I'm running out of time. 5 minutes. Okay, so salt. Salt in Australia is an issue. So you can see here that it's affecting 1 and 2 farms. This map is showing you soil constraints in Australia, and then if you overlay this with a wheat belt, you can see that there are real issues there. This work is at the moment led by Stuart Roy, but it has been, it's a long-term project, and Mark Tester probably is going to tell you more about this. So we find genetic diversity. So this is increasing salt levels, and then to the right you find varieties with increasing salt tolerance. So mapping of this QTL on material development is ongoing. But we do have already some quite interesting genes, and I like this because this is one of the few genes from Arabidopsis that really seems to be working. So when you put this gene into barley and you screen it in the field, under very high soil conditions, you see it's actually conferring tolerance. Similarly, we have another gene, ABP1. This is gene has been identified by Roberto Gaxiolo. It's also conferring tolerance to salt. It's a vacuolar pyrophosphatase, but it's actually one of the genes I find very promising because it enhances yield under control condition. It also works under P deficiency. So this is really a very interesting gene, and we are very pleased to have an IWIP project where we put the pistil 1 and ABP1 in wheat, and we pyramid those, and we have a third gene, the NAS gene, that enhances zinc and iron in the grain. So this has been funded this year, and I look forward to working on this project. The last trait I want to mention here is boron toxicity. This is also one of the really long-term projects of the ACPFG and other collaborators. And this is a tolerance gene that was identified in barley. So you can see here that Sahara and the high boron conditions is not showing those leaf bronzing and symptoms. And the trait is due to a duplication of a boron efflux transporter, BOT1, and you can see in the tolerant variety we have 4 copies of this gene as opposed to 1 in the intolerant one. And then in wheat, again, genetic diversity is there. So we have Holbert as the tolerant donor of this gene. The gene was cloned by Tim and his team. And the top one is actually the functional gene. You see it's a very complex gene. And in sequencing across other genotypes showed that this is truncation insertions in the promoter. All kinds of things happen to make this gene non-functional. A marker survey then showed that in the southern part of Australia, you actually have boron toxicity. So breeders, again, they have selected, so the red one is the functional gene, selected for this gene for the southern region, but have selected against it in the north because in that region you have boron deficiency. And that's one of the other issues I want to say because breeders really are reluctant to use this now because they cannot afford to have a disadvantage of a gene in any certain region. So one requirement really is that you have genes that have no disadvantage under normal conditions and also no yield penalty. So beautiful project, but probably not going to get very far. This is my second last slide. I just tried to, this is really a really rough one, and I need to think a little more about it. But what I'm trying to do is just to show you the pipeline. So if you do a forward genetics approach, you start off with identifying a tolerant genotype. You identify the QTL, you find MABIP, develop the markers, and then you need to validate it. So this already is a very optimistic schedule there. So it's actually rather twice as long. So the point I want to make is already after 3 years, usually funding is ending after 3 years, you just get stuck with the QTLs. And that's why there are so many papers out there, so many QTLs that never get anywhere because funding is just ceasing. So at some point then, the more reference genomes we have, the faster this will be. We get candidate gene list, and then you try to validate and do the science. So this is a 10-year, at least 10-year process. The project that I introduced to you are actually 20-year projects. So this is really too long. Maybe with GWAS, now we can be faster, and as I said, if we have, we can do this in the computer. I mean, this is a dream come true for me. So I think breeders become interested at this point, not so much before, so they want something validated, breeding ready. Again, a problem for researchers to get a project to that stage, that breeders really take it on, because they want to have a large effect, 5 to 10%, no yield penalty under control, as I said, effective in different genetic backgrounds, in different environments, and you name it. So really, there's a need to link up with breeding programs at an early stage to make sure this is getting, that they help us with that. I think industry then is more at the other end of the spectrum when they become interested in candidate gene list, but again, also there, major validated genes is what they love. And then they put it into their pipeline, test it as GM trait or as hybrid, as a delivery mechanism. There, the minimum requirement is 10% yield advantage under on average, because otherwise the investment is not really worth it. Again, effective in different genetic backgrounds and different environments. So this is a lot of things to do, from gene discovery to get it out there, and it's really something that anyone alone or any small research or universities really can't do. So we need to think about how we can better do this and have strategic partnerships to get this done. This is my last slide. This is one of a photo I took from our sub-1 breeders, or farmers actually, in India. So he was pretty happy and pleased with what he got there. And I think really the point I want to make is if we want to have projects like this and success and make a difference, we need more longer-term funding. We need to tap into this genetic diversity, and there are global efforts going on in wheat and rice as well to develop populations to get access to this gene pool. We need links with breeders and industry to help us get this done. And also, I would probably think if there were more researchers now moving from our Arabidopsis into relevant genotypes, that would help as well in the gene discovery world. And we need also to be specific about or strategic about the stresses we address. And I think drought, heat, P fixation, rather than P deficiency, but P fixation is a bigger issue, I think, and internal nutrient use efficiency. So enhancing internal nutrient use efficiencies to have plants that dilute without a yield penalty, be able to produce more with really less nutrients I think it's the way forward, especially for resource-poor countries. And this is my last slide. Thank you very much.
Questions for Dr. Heuer? Chair's prerogative on the first one. The phosphorus work in rice, is it a greater uptake of phosphorus, or does the gene actually allow more efficient use of phosphorus?
It's more uptake. It's just because the root surface area is larger, because it's growing better. It's better uptake.
So
yeah, so we have done 2 microarray studies to look into the starvation or the genes responsive. And it's usually not the starvation genes that we found. It's more the root growth and drought responsive genes.
Thank you.
Yeah.
Other questions from the audience? Yes, please.
So for the QTL study on the drought tolerance, how confident you are like QTL is stable? Do you have like a multi-environment or multiple testing about the different environment, the stability of those QTLs? For the drought tolerance, yes. You are showing how much you are confident about this QTL is stable in the wheat.
When we consider it stable.
So you have talked about the genes for the drought tolerance. So do you have a test or multiple environment which is proving that this is stable?
Yeah, so the graph I showed, this is actually, I think, 4 different sites. We also work with CIMMYT to test those materials in Obregon. And really the outcome of this testing was that it's a matter of deeper soil or shallow soil that is really determining if the alleles or the QTLs are effective or not.
All right.
And
thanks.
yeah.
Let's thank Dr. Heuer for an excellent talk. Our Our next presenter is Dr. Zahir Khan. You heard a little bit of pre-discussion this morning from John Pickett.